Welcome all. Welcome to my video. Hope you all are doing well. In this video lecture, I will be talking about cell cycle. And as we know, this cell cycle is a very important topic in biology. Okay, it's very important to have a clear concept on it for your further studies. Okay, so let's begin the topic. So when we talk about cell cycle, what is it? Okay, as we know, whenever a cell parent cell divides into two daughter cells, okay, it goes through various events. Okay, because as we know, during cell division there are certain cell growths takes place, then division is taking place. So it occurs with varieties of events, many events, and these events are what cell cycle. So one cell to the daughter cell, this occurs in a cyclic manner, okay, where varieties of events taking place, formation of raw materials taking place, DNA replication taking place, then divisions taking place of nuclear division, then cytoplasmic division taking place. So these things total we call it as cell cycle. Okay, so these events themselves under the genetic control. So when we talk about cell cycle, it is the sum of all the events by which a cell duplicates its genome and synthesizes cell content and eventually divides into two daughter cells. So it has two parts. Cell cycle has two parts. One is interphase and another is M phase. So let's come and talk about interphase. So when we talk about interphase, it is the period between the end of one cell division to the beginning of the next cell division. Okay, earlier this phase is known as resting phase. Actually, resting phase is completely a misnomer. It is not at all a resting phase. Rather, it is a highly active phase. It is very much metabolically active. Why it is said to be resting in terms of actual division, since no divisions are ever seen in it. We cannot see divisions in this phase. But what is happening in this phase? This phase is actually preparing itself for the upcoming cell divisions. Okay, for the next division, since the preparation is taking place in this phase, it is known as preparatory phase. Okay, so preparing itself is very important. Then growth change is taking place, then accumulation of certain raw materials taking place, which is very much required for the interphase. Okay, now when we talk about division, cell division, what actually happens? We see the normal basic concept. In case of human UPKC, we know this is a cell which contains the 46 chromosomes, right? Now when it will make its copies, okay, in the daughter cells, then it has to give complete information to the daughter cells, and that is it need to be give again 46 chromosomes, right? So in that reason, the duplication of genetic materials required. Okay, so that is what taking place during the cell division, that DNA replication. Okay, copying of DNA. Fine. So here it is what interphase. So when we talk about interphase, so see here G1 to G2. This complete phase is interphase, and then here is M phase. Again, M phase is divided into two further parts, karyokinesis and cytokinesis. So let's know about the interphase in this lecture. So when we talk about interphase, it has three important events: G1 phase, S phase, and G2 phase. Okay, so come up with the G1 phase. So what is happening in G1 phase? Actually, G1 phase is preparing itself for S phase. Now what is happening in S phase? S phase is happening the DNA replication. So in G1 phase, the cell has to be prepare itself for DNA replication. So what are the things, what are the steps taking place over here, what are the events taking place? First of all, since the DNA replication will take place, it requires a number of enzymes. The enzymes formation requires proteins. So here the protein synthesis will take place. So here the protein synthesis will take place. And in order to produce protein, the transcription processes or translation processes are important. These processes are important. So in order to carry out these processes, it required large number of RNAs. All kind of RNAs are formed here, whether it is rRNA, mRNA or tRNA. So RNAs are increased, RNA synthesis increased over here. Then what is happened? To carry out all these processes required huge number of energy. So lots and lots of ATP synthesis take place over here. And cell growth. So here growth occurs. Growth in the cell size increases. So here what is happening? Four major things. First of all, preparation of the raw materials required for the RNA replication, that are proteins. RNA for protein synthesis, lots amount of RNAs required, all type of RNAs, huge energy requires, and that's why ATP synthesis. And another event, that is growth. Cell will increase its size. So that's what happening in G1 phase. Now come in S phase. What is happening in S phase? Now see, already in G1 phase, everything that is required for all the raw materials required for the DNA replication is already prepared. That is, whether it's energy, it's RNA or DNA formation or enzymes, okay, RNA formation, enzymes, everything is there. So now what are the things happen? This is the first thing, DNA replication. Number two, centriole division. Now centrioles, we know it is present in animal cells, absent in plant cells. So centrioles will divide in order to division of the spindle fibers, right? So there is division of centrioles. In case of plants, there are certain factors that are helping in spindle fiber formation. But in animal cells, this centrioles division taking place in S phase. And the third thing which occurs, that is the formation of histone proteins. The formation of histone proteins. We know that these histone proteins, they are around it, the DNA threads are being wrapped around it, and it is forming the nucleosome. So this histone protein formation occurring in S phase. Now what happened? In a few books you will be finding that in G1 phase also histone proteins forms. So it's not wrong actually. What happened? In lower eukaryotes, 50% of the histone proteins formed in the G1 phase, and in higher eukaryotes, 80% formed in the S phase. So come over here again to what are happening in S phase. RNA synthesis, which is the number one event, then centriole divide in case of animal cells, and formation of histone proteins. Now come over here, what happened in G2 phase? Now G2 phase is preparing itself for the M phase. Okay, that M phase, what happened? The actual division taking place. So for the division, what are the things required? What are the things need to be happen? That preparation is taking place in G2. So what happening in G2 phase? So if we see the cells, so here here what are the things happen? Like the nucleus needs to be divided. So the cell have to prepare for its nuclear division. The organelles divide. Then formation, the very important thing, that is formation of spindle fibers. So spindle fiber formation, spindle fiber formation, the things which is required for spindle fiber formation, okay, occurs here. So this in order to form spindle fiber formation, it requires a protein which is known as tubulin. Okay, tubulin. So this tubulin formation taking place in G2 phase. Okay, then the formation of plasma membrane taking place, the formation of plasma membrane proteins, then taking huge amount of ATP required. ATP synthesis is very important over here. Now what happened now? Why ATP synthesis required? As we know in what happened in metaphase, we will be studying in future that in metaphase what happened now. This is the centriole, this is the kinetophore, and this is the chromosome. This is chromatin C. Okay. Now what happens? The spindle fibers will attach here, and it will attach and go to the poles of the cells. You have to separate and divide and go to the poles of the cells. Now what happened now? In order to separate one chromosome from another, one chromosome and spindle fibers, and to take it to the poles, separating one chromosome, it requires about 30 ATPs, okay, drops. So we can understand in case of human, we have to separate 46 pairs. So how many ATPs are required? So another is huge amount of ATP synthesis taking place, and again here, since proteins, tubulin proteins, the main other proteins require lots. That's why here RNA, all type of RNA synthesis also increases. So for M phase, okay, M phase, the preparation for the division phase taking place in G2. What are the major events taking place? The major events helping preparing for the spindle fiber formation, and for spindle fiber formation requires the tubulin protein which is being synthesized here, then plasma protein formation, then ATP synthesis increases, and as well RNA synthesis also increases. So this is what happening in all the phases of interphase. So here you will be getting all the points, what are the things happening during G phase or gap 1 phase, S phase, that is synthetic phase, why it is synthetic, synthesis taking place, no? And then G2 phase. In our next video, I will be talking about some important things like phases taking place in M phase. Then we'll talk about D0 or D0 phase. Thank you and stay tuned. Please subscribe my channel if you like the video. Like it. Please like my video and press the bell icon for my latest updates. Thank you and stay tuned.